This is session 6, soils. I would just like to note that session 6A has not been recorded, but we are now recording the next two presentations. I'm Caitlin Rose-Seiler, and I'm excited to call up our next presenter, Rachel Becknell. Rachel will be speaking about the role of soil microbes in tall grass prairie restoration, and she is joining us all the way from Washington University in St. Louis, Missouri. So I'd like to welcome Rachel. And Rachel, you'll just need to unmute yourself. Rachel, can you hear us? We seem to be having some technical difficulties. Just bear with us for a second. She's just having some issues unmuting, so just give us 1 second here. So Rachel, just on the AV pod underneath my webcam is how you'd unmute yourself.
Okay, I think we're good now.
Perfect.
All right, sorry about that, guys.
No problem. I'll let you take over from here.
I'm a little too used to Zoom now. All right. Hi, everyone. Thanks for the invitation. My name is Rachel Becknell. I'm a PhD candidate at Washington University in St. Louis, and I study the impacts of soil microbes on prairie plant species, as well as a rare and federally endangered plant species called Astragalus bibulatus. There'll be further information about my other research that I do, a link to a talk that I gave on YouTube at the end of this session. Okay, so really quick, I just wanted to talk about the motivation behind my research. I essentially want to utilize ecological theory to better inform the practice of ecological restoration, but in turn, to use restoration practice to inform ecological theory, to improve upon the theory. And this concept isn't new. Many of you probably already know Tony Bradshaw. He said back in 1983 that if we want to show that we truly understand how plants' ecosystems come together, we need to actually be able to do that ourselves in practice and make those ecosystems work. Right. So my experiment utilized 18 different tallgrass prairie species that are native here in Missouri. Okay. But first I wanted to go into a little bit of the theory behind the project that I did. The traditional explanation for how many different species can coexist in one place is that of niche partitioning. So if you look at this example, we have species 1, 2, and 3. Species 1 and 2 are more closely related to each other. They're in the aster family. And species 3 is more distantly related to those species. It's in the mint family. And because species 1 and 2 are more closely related to each other, they have a much more similar root structure than that of species 3. So species 1 and 2 probably are better at taking up soil nitrogen than species 3, whereas species 3 is probably better at taking up soil moisture than the other two species. So if you're looking at a situation where species 1 and 2 are attempting to coexist, and this is a very simplified example, these species are most likely not going to be able to coexist. One will outcompete the other because their niches overlap so much. Whereas if you had species 1 and 3 or species 2 and 3 together in one place, they're more likely to be able to coexist together because they fill different niches. More recently, we've been focusing on how soil microbial communities can also lead to coexistence. So if in this example, we have species 1 and 2, an aster and a mint, and species 1 has these soil microbial communities that specifically like to attack its roots, and oftentimes these are fungal pathogens. We have other species in the soil like viruses and bacteria that attack the roots of prairie plants, but fungi oftentimes are the ones that do the most damage. So you see around the roots of this adult plant that we have a high density of these red fungal pathogens. But the further away that you get from the plant and closer to the mint plant, you don't have as many of those. And the same is true for species 2. We have a lot of these green fungal pathogens that like to attack this mint species roots. But the farther away you get from that plant, the fewer you have of those pathogens. And this has an impact on the species offspring. So species 1, you'll see around the adult, you don't have very many of the seedlings from species 1 because those plants are being attacked by those fungal pathogens that are much more abundant around the adult. But the further away, these seedlings are able to escape those enemies and be able to grow closer to this mint species. And the same is true for species 2. You don't have very many seedlings of species 2 around the adult mint plant, but the further away you get, you have more of these seedlings. They're able to escape. And this is essentially what we call negative plant soil feedback, and just essentially where a plant species is much more negatively impacted by its own soil pathogens than it is by the soil pathogens of other species. So this is what should lead to coexistence of these two plant species over time. So in this example, we have two curves for these species. On the y-axis, we have the population density of both species. On the x-axis, we have the time. And you'll see that T1, which is time 2, species 1, its population density is much greater than that of species 2. So you'd assume that at this point, the soil pathogens around species 1 are also much more dense and having an impact on the offspring of that species. Whereas for species 2, because it's not very dense, its soil pathogens that like to attack its roots are also not as dense, and so its numbers are able to increase. So that at T2, you have a much more dense population of species 2 and far fewer species 1 because its offspring were being so heavily impacted. And so it's this fluctuation of the population density of these two species over time that should allow for them to be able to coexist. Okay, feel free to ask any further questions at the end, and I can definitely break this down a little bit more if you need me to. But getting back to the experiment that I performed, utilizing these 18 species, I wanted to explain my main objectives were to, one, look at whether these plant soil feedbacks could structure tallgrass prairie communities. And then understanding this could be very important in the field of ecological restoration. And second, I wanted to see whether the relatedness of these plant species could impact these, the strength of these plant soil feedbacks. And finally, I did a little bit of DNA work to look at the actual identities of the soil microbes that were affecting these species. Okay, so sorry it didn't come across here. This used to kind of break down in different steps when it was in PowerPoint. But essentially, in the first phase of this experiment, I collected soil from a reconstructed prairie at Shaw Nature Reserve that's run by the Missouri Botanical Garden here in St. Louis. And I added the soil to a sterilized soil mixture and planted seedlings of all 18 species in their own pots. And I allowed for them to grow for 9 months. And at the end of those 9 months, the soil microbial communities in those pots should be more species specific. You'll still have soil microbes that aren't species specific, but their numbers wouldn't be as great as those that like to attack the roots of these plant species or even potentially benefit these plant species. In the second phase of the experiment, in the main phase of the experiment, I took the soil from those pots in the first phase and also the fine roots, and I added them to new pots, and I put seedlings of each species in their own soil. But the important part was I looked also at the growth of each species in all 17 other species' soil. So that made for a really large experiment, but we also did this in sterile soil. And the reason we did it in sterile soil as well was we wanted to be able to tease apart whether it was actually soil microbes having any sort of impact on the results that we were going to see, or maybe if it was just maybe differences in soil nutrients as well. Okay, so the way that we calculate these feedbacks is by looking at how each species grows in their own soil compared to the growth of the other species' soil. And you'll see that in this example, the yellow species and the green species both grow worse whenever they're growing in their own soil compared to their growth in the other species' soil. And the way that we calculate this is by subtracting the growth of each species in the away soil from the home soil. So you see in this situation, we have a negative number, and this is what we call negative feedback, where each species is more negatively impacted by their own soil microbes than they are of the other species' soil microbes. So now to see if we actually saw this in my experiment, and in many situations, I did see this. This is Monarda fistulosa and Rudbeckia hirta. And you'll see that in the presence of soil microbes, each species grows relatively worse in their own soil than they do in the other species' soil. And this is these circumstances that we would expect to see for these two species to be able to coexist with each other. Now, when we look at these two species in sterile soil, this relationship completely disappeared, showing that it really was the soil microbial communities that was impacting the growth of these plant species. Okay, now looking overall at the growth of all 18 species, we're also able to look at the average feedbacks, and that's just taking an average of the growth, what we call the pairwise interactions, or just the growth of each species in the other species' soil. And you'll see, like on the far left there, AG, that's Andropogon girardii. And you'll see that most of these plant species are growing much worse when they're growing in their own soil than when they're growing in the other species' soil. Now, this is only in the presence of soil microbes. When you look at it in sterile soil, this relationship disappears. And that really shows how strongly these soil microbes are affecting the growth of these plant species. Okay. Next, I wanted to see whether these patterns that we're seeing could be impacted by plant evolutionary history, or how closely related these plant species pairs are to each other. Okay, so that interaction that we saw before is what we would expect if you were to have two distantly related plant species growing together. So in the case of the aster and the mint, we'd expect for this situation where I have distantly related plant species growing better in the other plant species soil. And that's because we expect for more closely related plant species to share similar soil pathogens. So if you have, see this orange species growing in the blue species soil and they're closely related to each other, they're not able to escape their soil pathogens because they share essentially the same soil pathogens. Okay, so if we were looking overall at the growth of these plant species, I would expect in live soil to see that whenever plant species pairs are more closely related to each other, to see those neutral feedbacks, to see really no difference in growth based on the soil that they're in. But then whenever more distantly related plant species are growing in each other's soil, I would expect them to be more negatively impacted by their own microbes and be able to escape their microbes by growing in the other species' soil. And I expect for this to only happen in the live soil and not the sterile soil. And this is essentially what I found in my experiment, which is a very exciting result. I found that those closely related species have much more neutral or weakly negative feedbacks. And then whenever they're more distantly related, they're much stronger negative feedbacks. So those plant species are better able to escape their own soil pathogens whenever they're growing in the soil of the other species. And this relationship disappeared in sterile soil. So this really is showing that it's being driven by the soil microbial communities. Okay, so finally, I wanted to look and see whether there actually was evidence for specificity in the soil fungal community. Because up until this point, we're only looking we're just looking at the responses of the plants, and we're not actually looking at the soil fungal community itself. Okay, so what I did was I collected soil from each plant species, extracted the DNA. This is pretty simplified explanation of what I did. But then we use these, what we call ITS primers, and it's like a barcode that specifically pulls out the DNA from fungi, which is they are the species that we wanted to look at. And then I performed some bioinformatics and statistics to see whether these microbial communities were more similar. Okay, and I wanted to see whether there was a pattern of specificity and association. So this next figure ended up getting a lot more jumbled than I would have hoped. It was a figure that should have been like stepping through, but again, it was in PowerPoint. The way that you look at a figure like this is essentially the dots that are more close together are the ones that are more similar to each other. So you see this top dot up here. Let me move my mouse so you can see it. You'll see that agar, Andropogon gerardii, its soil communities, they're clustering together, so they're more similar. And you see by all the little ovals that I have all over the screen that most of these plant species, their soil microbial communities are much more similar to each other. And so like you see agar, those are 3 separate samples that I took to compare to each other. So that was a pretty exciting result there. And I can explain this a little more detail if you all would like in the questions. So essentially, I ended up finding that there is strong evidence these negative plant soil feedbacks that I see in my experiment are prevalent among these tallgrass prairie plant species. The next step would be to take this experiment out into the field and see whether we end up seeing similar results. And I think that a really important step would just be to perform a tallgrass prairie restoration experiment where we're looking at these plant soil feedbacks. So I also found this strong phylogenetic signature with more distantly related plant species having stronger negative feedbacks. And I think that in restoration, this could be a really important finding, especially if you have plant species that that are hard to establish in your restoration. Maybe you have like a Asclepias species, a milkweed species that you're having problems getting to grow. Well, maybe you don't want to be growing it next to other Asclepias species. Whereas if you end up to find positive feedback that this plant species shares beneficial soil microbes with other milkweed, then yeah, maybe you do want to be growing these plant species next to each other. So it's really important information to have. And I found that they do possess distinct soil fungal communities. And I do believe that incorporating these native soil microbes into restorations could be really useful for having greater coexistence and increasing the biodiversity in your restorations. And I should have added another slide on here. I did want to add one caveat, is that you have to be really careful whenever you utilize soil microbes in your restorations. One, if you were to take a lot of soil from a remnant prairie, and I think that you all already know this, if you were to do that, then you could really negatively impact this remnant community. The one thing that you could do is that you could inoculate seedlings in the greenhouse with these soil microbial communities. And what we found is it really doesn't take very much soil inoculum at all to end up seeing a positive response. So that's an option. I've found that in my experiments where I'm looking at the impacts of soil microbes on rare endangered plant species, that this is the case, and it's actually better to attempt to inoculate them in the greenhouse and let them get used to these soil communities before you try to reintroduce them. And finally, a lot of people are trying to use these commercial inocula that you can get online, and oftentimes they have invasive mycorrhizal species in them. And I know we already have enough problems with invasive plant species. We definitely don't want to create a larger problem by having invasive mycorrhizal species taking over in the soil as well. So those are just things to keep in mind. And I think we still have another 7 minutes to chat. So please feel free to ask some questions.
Thank you so much, Rachel, for the awesome presentation. You've got some really beautiful pictures. So thank you for that. And to our attendees out there, if you have any questions, feel free to type it in. I can see we've got a number of people typing in right now. So I will just pull up some of our questions. The first one is from a listener named Jessica. Do you know of any studies looking at microbes for natives versus invasives?
Yeah, this is actually becoming a lot more popular. It's not something that I've been looking at yet, but I was just like I was talking about how commercial inocula can add these invasive mycorrhizal fungi to the soil. I know that there are a number of studies out there finding that using these inocula with these invasive mycorrhizae actually will facilitate the establishment of invasive plant species. So that's definitely not something that we want. And I know that there's a whole wealth of information out there. So if you're looking for this literature and you don't have access to it, feel free to reach out to me, send me an email, and I'm happy to send you papers on it if you would like to look at it.
Her name's Sarah would like to know more about your research on rare species. Is that project ongoing, or have you published any part of it yet?
Yeah, actually, that paper was just accepted a couple weeks ago, and it should be published in Restoration Ecology probably within the next month. So keep an eye out for it. If you'd like, feel free to email me, and I'd be happy to send you an advanced copy of it. And you should have the link for my talk that I have on YouTube. I do go into that particular project towards the end of my talk. But really quickly, I was looking at the growth of this endangered legume species because it only has 8 populations left on the planet. And we're trying to understand why we can't get it to grow in our reintroductions, despite trying to match the ecosystem as much as we can to the remnant ecosystem. And what we actually found was really cool is that it grows much, much bigger and much better whenever it's growing in the soil from these ecosystems where it's already growing compared to where we want to reintroduce it. And what was crazy, you saw how I did the live and the sterile comparisons in my experiment. When we had this legume growing in the soil from the place where we want to reintroduce it, it grew the same size as whenever there were no microbes in the soil at all. So it's like these glades just do not have the soil microbes that it needs. And we need to do something to add these soil microbes. We think it's the rhizobia, those bacteria that infect the root nodules. It's really exciting. I'm doing some DNA work on it right now to try to identify the microbes. That'll be another paper that'll be coming out.
Very exciting indeed, and it's really exciting to hear about your upcoming publication. I think we're going to have the URL, the link to your YouTube video that you just referred to, that'll be available in the lobby after this presentation. Um, we have another question from a listener named May, and it's actually been seconded by somebody else, by Ashley. So May says, based on your results, post-specific, if we are wild harvesting seed, is there also value in wild harvesting soil for inoculating the planting with these specific soil microbes? I can do this with low impact with a 16 millimeter diameter soil probe.
Yeah, definitely. I think that that would be the least invasive method for doing it. It's actually something that agriculture companies like Monsanto, or what is it, Bayer now or whatever, they're actually looking at doing this technology where they're coating their seeds in soil microbes before they plant them. So you'll be doing that essentially by taking the inoculum and planting your wild harvested seeds with it in your restoration. It's a great idea.
Thank you. Yeah, thank you. Thanks for answering that question. There's actually a few more people that were looking for that same answer. So that's really good to know for our group here. We have another question. How would you ensure your microbial source is legit and not risk inoculating your soil with are there known go-to providers?
Yes. Well, let me pull it up here for you real quick. You can actually find, oh, I know what it's called. It's called MicroBloom. So right now, this company is actually run by researchers who have recognized the problem with these microbial sources not being legit, as you said. And they actually started cultivating their own native soil microbes to be able to distribute to people. You can get it off of Amazon, actually, MicroBloom, M-Y-C-O Bloom. And she sells them in really big vats. And I personally know these people, and I know that they take great care whenever they are culturing these microbes. So unfortunately, I haven't heard of anyone doing this in Canada, and her company is the only one I've heard of doing it so far. But if you all want to start a business, I think that it's a great thing to do. And I think each region really does need to have someone making, having native mycorrhizae and other species for restorations so that people aren't going to these commercial inocula.
Good opportunity for someone. Last question really quick, if that's okay. Should we inoculate the soil post-seedling establishment?
You can definitely do that. I've seen a couple papers where the researchers actually took the soil and they kind of like broadcasted it out like they would seeds after the seeds had been planted. I would have thought that it would have dried up or not been beneficial. They did end up seeing positive impacts. I think it would be probably good to do that, you know, not when it's like about to rain or something, because you don't want for it just to completely like soak through and disappear. But I think that would be a good idea. Like if you've already established it, it's worth a try to see if it would be beneficial. And the scientist in me wants you to kind of only do it on maybe half and compare the other half of your restoration. But yeah.
Sounds like a good project or an experiment for someone, that's for sure.
Yeah.
Yeah, well, that's all the time that we have today. So thank you so much, Rachel, for sharing your experiment with us. I know there's a few more questions we didn't have time for. If you do have time, you're welcome to reply in the chat box there. And I'd like to call up our next presenter. So our final presentation of the afternoon is about phytotechnologies for remediation and reclamation of high salinity soil. And I'd like to call up Liz Murray from Earth Master Environmental Strategies, and Earth Master is one of the bronze sponsors of this event. Liz also did a case study earlier today, so I hope some of you were able to catch that. And with that, I'll turn it over to you, Liz.
Great, thanks for having me. I assume you can hear me okay?
Yes, perfect, loud and clear.
Great. Yeah, so those of you who are able to attend the case study, a couple of these slides will be familiar to you, but I'll present a bit more detail on the topics as I go through this. Just a note on the previous speaker, adding bacteria to seeds and soil. That is a regulated activity in Canada. You need CFIA approval to do that. So if you are purchasing products from the internet, make sure that they are approved for use in Canada, because you could create quite a bit of problems if you are using products that do not have CFIA approval. All right, so I'm going to be talking a little bit about phytotechnologies for the remediation and reclamation of high salinity soil. And this is a topic that the company that I work for is trying to develop more technology around. So I work for a company called Earthmaster Environmental Strategies, and we're a Calgary-based environmental company. We do all sorts of environmental services, but we also have a technology that is focused on phytoremediation. We call that technology PEP systems, and we use it to treat contaminated soil in an eco-friendly and responsible manner. So we know plants are capable of doing a lot of really cool things, and two of the things that they do we like to use for remediating soil. So the first is phytoextraction, or the ability to take up salts and trace metals into their above ground tissues, and also phytostimulation, where they have a bacterial colony around their roots, and those bacteria are able to use hydrocarbon contamination as a food source. And that means that the bacteria population can break down those hydrocarbons into non-toxic materials. We're going to talk about phytoextraction today. So it's great that plants can do these things, but often when we're looking at contaminated sites, we're looking at poor quality subsoil, and it's really challenging to get the plants to grow. So we've come up with a technology to address that issue. And we call it PEP systems, which is plant growth promoting rhizobacteria enhanced phytoremediation systems. And this is a technology that we co-developed with Dr. Bruce Greenberg from the University of Waterloo. He's now retired, so we've taken over that technology and all the research and development around that technology now. So everything is now based in our Calgary labs. So PGPR, what we do with that is we actually coat it onto the plant seeds. And then when those seeds germinate, they colonize the roots. And those plant growth promoting bacteria provide the plant with hormones to stimulate plant growth, as well as molecules that reduce the stress ethylene the plant produces. So the combination of promoting plant growth through hormones and reducing stress ethylene allows these plants to grow in the very difficult conditions that they find with challenging soil contaminations. So I'm going to look at several things in this talk. So we're going to look at a case study using PGPR in the prairie salt remediation setting. So this was the case study that we presented at lunchtime, so may be familiar to a couple of you. We're going to look at lab studies that look at ways to increase biomass when there's situations of increased salinity in the soil. How do we make our phytoremediation system better? Then we'll look at lab studies that look at the application of PGPR with grass plugs and native species of grass plugs. Then we'll look at some follow-up field studies with that particular PGPR grass plug combination, then very briefly touch on some wildflower work. So this is what we know about phytoremediation with salt contamination now. And this was a field application that we did probably around, I don't know, 7 years ago or so. The goal was, this was a site in southern Saskatchewan, and the goal was to revegetate the grassland site to at least 70% of background levels. And by doing that, reduce soil salt levels over time, which will allow for sustainable plant growth. So we actually deployed this in the summer of 2010. And we used a series of 4 species of plants and a Pseudomonas. And then we managed these treated soils over time. And just very briefly, what we found is this is what the site looked like when we got there. It had ECs of up to 40. It had parts of the area that no plants could grow on, and it was really challenging for them to get a revegetation going. That's a groundwater collection system in the background that they had a lot of trouble maintaining and keeping working. So we deployed bacteria-treated seed, and this is what we saw after 3 months of seeding. So we were able to get quite good establishment of the grass. And this is what we saw in year 2. So that's what we know about salt remediation with our system now. We know we can decrease ECs 1 to 2 decisiemens per meter, or per meter. That we can take up sodium and chloride to an amount of 150 kilograms per kilogram. And we also know that that is fixed per gram dry mass of the plant, and it is plant specific. So one of the challenges we have with getting this commercially deployed is it takes a long time to remediate salts using plants. And so we're looking at ways of trying to make this whole system faster. And so these are some experiments we've gone through to look at how do we increase the amount of salt remediation using plants. This is some historical data from Bruce Greenberg's lab. And basically what it says is whether you have a medium salt soil or a high salt soil, the amount of sodium and chloride that the plant takes up is going to be the same. And it doesn't matter whether the plant is treated with PGPR or not, it's going to have a set capacity for salt uptake. So PGPR does not influence the amount of salt the plant can uptake per gram of dry mass tissue. What it does do is it increases the amount of plant biomass in the soil. So if you have an increase in biomass, you will automatically get an increase in remediation of salt, or more salt taken up in the above ground material. So we did some lab studies using produced water, and this is produced water we got from 2 different well sites in Alberta, and you can see the parameters of that produced water, quite high in chloride, quite high in sodium. The EC is insane, and the SARs are insane as well. So quite challenging conditions to grow in. And this is what we did. We set up some germination studies. We were looking at the 4 individual species that we use on remediation sites. And those species are selected because they have a lot of root biomass and they have a lot of above ground biomass. We looked at 20 seeds per plate. We had plus or minus PGPR and plus or minus other additives. And we just grew these in varying ranges of produced water and looked at them for 14 days. And so this is the kind of information we were getting from these plates. So I'm just going to walk you through this. We have 4 species of plants: annual ryegrass, perennial ryegrass, tall fescue, and tall wheatgrass. The numbers across the top of the graph there are percent produced water. So it went from 0 to 100%. And you can tell by looking at, so everything of the same color is in the same amount of produced water. And it's just the different squares and circles and triangles that distinguish the different seed types. So basically, what we were looking at is growth curves over a range of produced water. This happens to be untreated seed in a range of produced waters. We were looking at the lethal concentration, which is the concentration that's required to kill 50% of the population or prevent germination. We're also looking at mean germination time, as well as percent germination out of the 20 seeds that we planted. And just to make it a little simpler, once again, we're looking at untreated seed. If you look at ryegrass versus tall fescue, you can see that as you increase the amount of produced water, approach the purple here, then you inhibit germination and growth of the seeds. What happens when you coat these seeds in PGPR before you expose them to produced water? So for this particular graph, you're looking at the solid lines versus the dotted lines. The seed that's untreated versus the seed that is treated. And the particular bacteria we're using we call CMH3, which was isolated from southern Saskatchewan. And you can see in all cases that when you have PGPR coated onto the seed, that it increases the amount of germination in produced water exposure. And I've actually converted the produced water to milligrams per liter of chloride and sodium. You can see at the top there. So there's an obvious advantage to coating these seeds in PGPR and then when they're germinating in high salinity conditions. I do want to just point out here that PGPR offer an advantage to seed germination when they have challenging conditions. If you're growing in optimal conditions, you won't see an effect from coating with PGPR. It really shines when you have challenging conditions like high salinity or hydrocarbon there or something along those lines. So how do you quantify this result? We look at mean germination time. So all of the hatched bars here are untreated controls, and you want to compare them to the same color of the solid bar. So basically a hatched green is an untreated control, and the solid green is what happens when you coat it with bacteria. And we're looking at individual species of seed here. So ARG, PRG, tall fescue, tall wheatgrass. And two different concentrations of chloride in this particular graph. So what you want to see is you want to see a decrease in mean germination time compared to the untreated control. So you want the solid bars to be smaller than the hatched bars for the corresponding color. And when you look at percent germination, you want that to be bigger. So you want the solid bars to be bigger than the hatched bars for the corresponding colors. And I've put that in chart form below. So basically what we want to see is PGPR decrease mean germination time and increase percent germination. What does it do with LC50, or the lethal concentration? And these are the two different produced waters that I showed you the parameters for at the start. Once again, anything that's got a hatch in it, that's an untreated control, and you're comparing it to the corresponding solid bar. So this is expressed as a milligrams per liter of chloride in the produced water. And what you want to see is a larger tolerance to the chloride. So you want the bars to be bigger to show a positive effect of the bacteria. So in this case, you can see that the solid bars are much bigger than the hatched bars. And so the bacteria provide a protective effect from the lethality of higher concentrations of the molecules that are in produced water. So what we wanted to do was look at some additives to see if there was a way that we could improve biomass production. And this would be useful in high salt conditions, not only for remediation, but also if you're looking to revegetate a particular area that had elevated salinity, then you could look at using a bacteria to give it a better chance of establishment and survival. So we're looking at salt remediation through plant uptake. We know that it's fixed, and we know that the only way that we can increase remediation through plants is by increasing biomass. So the first thing we looked at was a plant hormone called gibberellin. So we added gibberellin into our seeds. It's a chemical messenger that breaks seed dormancy, and it promotes germination and stem growth. And so what we wanted to see was, could we improve germination by the inclusion of gibberellin? And we looked at it, once again, hatched bars being untreated, and compare it to the solid bars. We looked at PGPR alone, or gibberellin alone, and then in combination. And we're looking for an improvement over just the bacteria alone. And if you look at this and compare the solid bars to the hatch bar with LC50, once again, we want to see that the plant or the seedling can tolerate a larger concentration of chloride, so has a bigger bar than at the untreated controls. And you can see, if you compare gibberellin to just the bacteria alone, it doesn't really offer a lot. It might be for a specific seed type, like tall fescue, you might see a bit of an advantage with gibberellin. But when you look at the seed mixtures like we use, there isn't anything that's going to give a real advantage that would make it worth the added cost to add it into a seed treatment. If you look at mean germination time as well as percent germination, once again, you're looking at the untreated control versus the treated control. And looking at the averages, you want to see a bigger increase in percent germination and a smaller amount on the mean germination time. And really, there isn't any advantage to adding gibberellin over just using the bacteria alone. So we kind of struck out with that one. Next, we looked at tryptophan, which is a precursor to one of the plant hormones. And we did the same sorts of experiments. Can we increase a tolerance for chloride by treating the seed with tryptophan? And once again, we did it with bacteria, and we didn't do it on its own. We did it bacteria alone compared to in combination. And once again, compared to the hatch control, which is untreated. And we're really looking for an improved tolerance to chloride for an LC50 And we're looking for an advantage such that it would be worth it to add that extra step in treating seed. And what we saw was that compared to bacteria alone, adding in a tryptophan really didn't make that much difference for tolerance to chloride. And then if you look at that with percent germination and mean germination time, once again, there was no advantage to including L-tryptophan in the seed treatments. So next we looked at adding in some different bacterias, and we did that by just plating some soil bacteria, looking for interesting morphologies, and then identifying them using 16S RNA, and then adding those into the seed treatment. And what we found, once again, untreated control, and the solids are the treated control. We had our standard CMH3 bacteria, and then we're looking at an isolate called I3 and then another one called SR to see if we could actually get something that's better than CMH3. And we did not see an improvement in the tolerance of chloride for the LC50 compared to what we already had. And when you look at the percent germination and the mean germination time, there really wasn't any improvement over what we already had. So we decided that on their own, there wasn't anything that we discovered that was better than what we already had. The question that remains is, can they be used in combination? Because they do have different modes of action. And those studies are ongoing to see if we can improve plant biomass using combinations of bacteria. So I'm going to switch to some native grass plugs that we did a few laboratory and a few field studies with and looked at some reclamation applications. So these are environmental growth chamber studies, and we were using a salt-contaminated soil from southern Saskatchewan, and you can see the parameters of it there with the salt or chloride concentrations and the sodium concentrations, and you see around 17. So we were asking the question, can PGPR be used to facilitate plug establishment in challenging conditions? And so this is how we did that. We got some native prairie grass plugs, in this case, blue grama grass, and we were treating them with PGPR slurry. And we actually added this slurry directly to the root plug. And then we potted them in the elevated salinity soil. And the reason we did that is because we wanted to elicit a stress response in these plugs that would be similar to what they encountered when or trigger the same stress response as what they would encounter if we were planting in drought conditions and without the salt stress. We didn't fertilize, we maintained the moisture levels, and we monitored these for 7 weeks. And I just want to stress these are a small number preliminary studies, but they were quite encouraging. So we were looking at a plant height measurement as an idea of whether we would be able to increase biomass. We had 2 different concentrations or amounts of PGPR, and you can see that there's a definite advantage to growing with 10 mils of PGPR added to the plugs when they were planted in the high salinity soil. And that's what they look like as they grew. So we ended up taking the information from that preliminary lab study and applying it to a small field study, a little proof of concept study. What's not shown here is that we also included L-tryptophan in these combinations, and there was no advantage to including an L-tryptophan in this. So with a preliminary field trial, we had several trial locations located southeast of Brooks, and these were reclamation projects at former oil and gas sites in native prairie setting. The plugs were planted near the end of June, and the areas were overseeded with a native range mix for client purposes. And we ended up working on sites that looked like this. So they had areas that just would not revegetate. And so we set up some plugs in those bare areas, and this is what we saw with looking at height. So we're comparing purple to green, which is treated versus control or untreated, and we're comparing maximum height, which is the light colored, and median height, which is the darker colors. And we had 3 treatment areas, a small number of plugs on each treatment area, and the chart on the side will just give you the nutrient status of those areas that we were planting into, so just to control for the soil that we were planting into. And what we found is that it looks like treating with PGPR when you plant these plugs does give them an advantage as far as growth of the plug. And this is some not great pictures of what they looked like. Like I said, this is a small trial with a small number of plugs. And we were looking at a visual evaluation of plant health, whether it was poor or good. And we did see visually that there was quite a better outcome when these plugs were treated with P2PR before they went in the ground. And so that was our preliminary field trial with P2PR treated plugs. And this is just a quick overview of an industrial site we had here in Calgary, where we were doing some wildflower trials. And this area was previously used as a farmer market, then this for storage of equipment. You can see it's got a bit of a red tinge to it. It was covered in at one time with that red crushed gravel material that was used for making roads and that kind of thing. The soil was extremely hard packed, very poor quality. And we set up several field trials with plus and minus P2PR treated seed. Some involve grasses, some involve seed mixes, and some involve clovers. I'm only going to talk about the seed mix ones that had wildflowers in them. And the areas were seeded at a very challenging time. So in July, and with no soil preparation. So we basically deployed the seed, just lightly raked in the surface, not that you could really call it raking given that it was hard as rock. And then we watched. And this is what those sites look like. So we have the two sites, one that was a bit more protected and one that was a bit more exposed. Just a note here, the reason they look so bare is that we had a bobcat go over these sites and take off the weeds before we deployed the seed. So that's why they look so pristine. And these are the seed mixes we use. So one was, we have a collaborator that we're working with that's establishing a compost facility there. So the Highfield mix was their choice. You can see it's got a bit of a blue tinge. CFIA requires that when you put a microbe on seed, that it has a dye so that you know the seed has been manipulated. You also need approval from CFIA to put microbes on seed and then put them in the soil. So yeah, you need to make sure that you're following the rules when you do this kind of thing. And then the pollinator mix on the other side, that was one that our Northern Operations guy, Ben, put together. He's a bee guy. And so he put together a series of flower mixes that would promote pollination. And it was a pretty challenging conditions. And I'm just going to give you a quick overview of the end of the season pictures. And what you can see, untreated meaning they didn't have P2PR, and then treated were seeds treated with P2PR. You can see it's pretty sparse on all of them, but what did grow, we saw growth. Where we saw no growth on the untreated sites, we saw some growth on the treated sites. And not that you can see a lot here, but we did get some plant growth from the seeds that we put in. And then if you look at the pollinator species, you can see once again it's pretty bare in the untreated controls. We got a little bit of growth in the areas where the seeds were treated. We did two types of treatment with the pollinator species. We actually treated the seed, and then we, in another case, actually sprayed the bacteria on the ground and then deployed untreated seed and raked it in. So we did a couple of different things there. And just a quick summary on what we found. The conditions were really extreme for this trial. The ground was really hard, and we planted in July. As soon after we planted, we had some pretty bad drought conditions. We did get germination and then a pretty significant drought, and then the heat resulted in plant death, which made it almost impossible to try and identify what did sprout and then die. The weeds did come back, and they were a bit of a problem. We thought we could identify what they were, so we think we knew what was what we planted versus what took over. For the Highfield mix, actually radish, kale, and turnip did pretty good. We're kind of surprised by that. We saw some vetch, some grass, and some Phacelia in that, and possibly some other species there, not 100% sure. With the pollinator mix, we did see California poppy pop up quite a bit, and we did see some cornflower. The rest we found were really hard to evaluate, especially at the end of the season. I think the other challenging thing we face in that site is there are lots of critters on that site, and so critters like to sample some of the plantings. So it was a challenging site to work on, but it gave us some ideas that we could probably get some success working more with some wildflowers in some of the trial sites. So that's a picture of some of the wildflowers we did get. So at least we had a tiny bit of color on the site, which was great. So what's the next steps? As I mentioned, we're going to start combining PGPR. PGPR is an interesting world when you're coating on seed. More is not better. And I think Rachel mentioned this, that you can inhibit plant growth if you have the wrong kinds of bacteria. And in this case, we're finding if you have too much bacteria on the seed, you will inhibit germination. So you need to do the trials to find out what the right amount of bacteria is for what you're working with. And so we're looking at doing some PGPR combination studies, both with rooting approaches, growth chamber studies, and some field deployment studies, as well as some additional lab studies for the grass plugs, and hopefully some more field trials with those grass plugs to do more enhanced reclamation studies. And I just want to finish by thanking the National Research Council, because they've provided quite a bit of money to help fund some of these studies. And also I want to thank the clients that allowed us to do some of these trials on their sites. So I'm happy to take any questions.
Thank you so much. That was a really, really detailed presentation. So thank you for your time and sharing all of your research today. We have quite a few questions from our listeners here. We'll start with one from Melissa. Oh, I guess I have to make it visible. There we go. Is there any research into whether or not NaCl is re-released into the soil through plant death/breakdown after phytoremediation?
So if you did not remove the above ground plant biomass from the site at the end of the growing season, then yes, that sodium and chloride would go back into the soil as the plant degrades. So one of the reasons why we choose to work with the grass species that we use is that we can use just standard agricultural equipment and harvest them like hay. And then that actually removes the sodium and chloride from the site once the plants are harvested. So that's the way you actually remove it from the site.
Thanks. Rennie is wondering if there's a reason why wildflowers not native to the region were used in the trials. Locally sourced species may be more suited to these conditions than those from the US. Do you have any comments about that?
So what we were looking at is pollinator species, and Ben chose those based on providing pollinating species for bees. So Calgary's a bee city, and so there's a big promotion of trying to get more bee-friendly plantings, and so that's partly the reason why. I can't completely speak for Ben on the other reasons why he did this. Everything was sourced through Pick Seed, I believe. So that's where we got all that seed. I don't know if Kevin's attending. If he wants to put something in the chat about that. I think Ben used Pick Seed to get these, if I remember correctly.
Thanks. Patricia is wondering if bees nest in saline sites, could maybe leaving bare saline sites in prairies? Do you have any comments?
I am not a bee person, so I am not going to even attempt to answer that question. So if there's a bee person online that can put something in the chat, that's a great question. I'm going to ask Ben that question when I see him and see if he knows the answers to that.
That is a great question. And May is wondering if you ever give this presentation to farmers who have natural salinity issues. She acknowledges it's a different kind of salt. Or have you had any farmers approach you with interest in this product?
So the bacteria combination that we use is approved by the CFIA, Canadian Food Inspection Agency, for use in remediation. There are versions of what we use that have been approved for use in Canada for agricultural purposes. So yes, there are lots of products out there as inoculants that treat seed with bacteria for agricultural purposes. They've been vetted by CFIA and approved by CFIA. So make sure if you're going to go that route that you look for CFIA approval. Kevin, I notice you're on there. So he can maybe chat a little bit about what the farmers are looking for. There are a lot of salt tolerant species available, anything from, I think, alfalfa to wheatgrasses, that kind of thing. So that has been developed for farming applications. The one thing that we look for, which I mentioned in the case study we're talking about, there are different ways for a plant to be tolerant to saline conditions. One is that they just accumulate the salts and deal with it that way. The other is that they have salt glands. And so as they take up the salts, they just excrete it onto their tissue. And then when critters walk by and jostle the leaves or it rains, that salt just falls back onto the ground and goes back into the soil. You don't want to use those kinds of plants if you're looking for remediation. You want to use the kind that accumulate the salts, and then you can remove it from the site that way. But I know Kevin has several salt-tolerant species available, and he can probably speak more to what farmers are looking for and how much he's dealt with farmers. But like I said, the approval for our product is for remediation and some reclamation, not for agricultural purposes.
Thanks for clarifying that.
Yeah.
I think this will be your last question since we're a little bit over time. And Stephanie is wondering about, in reference to the case study you mentioned at the beginning of the talk, what was done with the plant biomass afterwards? Was it baled and removed every year? Could the removed vegetation be used for livestock, or would salt be a concern for livestock?
Yeah, I can't recall offhand what the biomass was on these sites. Can't remember. Was a bale? Yes, it was bale and removed every year. We do not, well, it's up to the client what they want done with that vegetation. Because it's oil and gas related, they ask that it not be shared for livestock use. The reason being is that although the toxicology studies have been done on it and the amount of salts in these tissues are not a big deal, if something were to happen to the beloved cow because they ate grass that came from a oil and gas site that was being remediated, the optics just don't look great, and the clients never want to take that risk that it may be completely unrelated, but people are going to automatically draw that conclusion, so they just don't go there. But I can tell you that when we work up north with these for salt sites, the muskox love to eat it, and there's lots of critters that love to eat it. And yes, it's slightly elevated. It is slightly elevated in salt, and the critters find it yummy.
Well, thanks for that answer. That's all the time that we have today. And I do want to mention that Sarah Semler has put in some information about alkali bees, and she thinks there are desert species of bees, and she also put in a link too. So anyone who's interested more in bees can look there. So that's kind of the end of our session 6 about soils. For those of you who have not had enough about soils, I would like to encourage you to head over to the discussion group about soils with Leanne Chai and Sarah Thacker. We also have an open discussion group going on too. And if you haven't made it to the exhibit hall, you've got to head over there and check it out. We've got some really awesome booths. There's some really great information in the poster session too, so be sure to check that out at some point this week. You can download the PDF or the slide deck, or even watch a video as well, maybe while you eat lunch or have a coffee break. So with that, thank you very much, everyone, and we'll see you on Feed Loop in the exhibit hall or networking. Have a great rest of your day.